Hi, I'll be talking about discovering biomarkers for pediatric acute myeloid leukemia using single-cell RNA sequencing. A brief introduction about FLT3 Itd Aml, the specific type of cancer that I work in. It has a 31% 4-year progression-free survival, which is okay. There is a lower bound and upper bound that we can work with, but which means it's a good spot for biomarkers to kind of be important. But currently, the current biomarkers are insufficient to predict outcomes. And over the past few years, there's been few new therapies. So stratifying patients may better use current therapies and will help existing patients or new patients figure out kind of like what they're looking at, because it can be a stressful time. So what we did is we used 10x Genomics single cell platform on bulk AML cells, so the CD34, CD38 positive cells. We did a standard processing with Seurat, SCT transform, and Harmony for batch correction. We then did some filtering on the clusters to select clusters that most impacted survival. We used an in silico deconvolution tool to deconvolute the clusters from a single cell RNA-seq to bulk RNA-seq, and then looked at some pathway analyses. So looking at like a 10,000-foot view at clusters, here we have the favorable patients that did not relapse, poor patients which did. You can already see that there's a big difference between some of the clusters, where clusters don't show up in the poor patients, but they're very enriched in the favorable patients, and vice versa. Looking at different statistical tests, the chi-square test, we see the same kind of thing with a few different clusters, where some clusters that have a big difference in frequency between the poor and favorable outcome groups, you see several clusters that also are significant based on event-free survival. So taking these clusters that we're finding and trying to figure out how much do they impact survival using existing bulk data. So we took the single cell clusters that we got earlier, deconvoluted them, and trained a lasso regression model on a split training test cohort of FLT3 Itd Aml. Here I'm just showing you the testing cohort to save space. And you can see that there is a difference in survival, and by the end, and it's about 25% of an effect size. Looking at the FLT3 Itd negative patients, you do still see that there is a significant difference, and there is an effect size, but it is smaller. And one reason why I think it's so significant is just because the difference, comparing the two, the N is much higher. In figure B, we did the same thing. However, we subset the data to only include FLT3 Itd patients when we did the clustering, excluding the few CBPA patients that we have a different mutation that also negatively affects survival. And we see pretty much the same thing. You see a good effect size and and it is significant over time, and then a smaller effect size in the FLT3 Itd negative patients with, in this case, actually a similar p-value. It's not as low. So then we wanted to see what makes these clusters special. So we first looked in the FLT3 Itd and CEBPA clusters, and we saw that in a cluster that increases event-free survival, mTOR signaling pathways decreased, and glycine, serine, and threonine, and nitrogen metabolism are both increased. And in a separate cluster that also increases event-free survival, however, this cluster was done in the FLT3 Itd only cluster, so the second survival analysis that I showed. You see mTORC1 signaling is decreased, and you also see adipogenesis, glycolysis, fatty acid metabolism oxidative phosphorylation and reactive oxygen pathways are all decreased. Now, kind of circling back and looking at the immune system in the FLT3 ITD plus CBBA, again, the first survival analysis I showed, you can see that in a cluster that decreases event-free survival, we see an increase in tolerance, and opposite in a cluster that increases event-free survival, we see positive, I checked, regulation of T-cell co-stimulation, Go terms. Then what we have been experimenting with is a cell-to-cell communication analysis tool. And here is a Pca loading plot, and you can see an increase in variance in CD27 and CD70, which kind of might illuminate that we're looking at some kind of immune cell interaction, but we're not capturing the whole thing because we're only looking at the cancer cells. We're not looking at the immune cells. So overall general conclusions, deconvolution can train survival models pretty effectively, and including CBPA patients might prevent overfitting. However, I haven't confirmed this yet. And one thing I didn't show is this is more consistent than an existing model that was developed in 2019, 2020, the PLSC6 model, where I couldn't easily detect it, and it didn't show much significance. What I'm looking to do next is assessing training on overall survival, because up until now I've just been doing event-free survival, and continue expanding on what I've just showed in figure 4, looking more pathways and trying to figure out what's special about these clusters and how they're exactly affecting event-free survival. I'd like to acknowledge all of the cores, both here at Case and at Cleveland Clinic, that have helped, including all the members of the Wald and Huang labs, all of the publicly available data that I've used, and the Children's Oncology Group, especially for providing the samples.